Okay. Hello, everyone. I'm very glad to have this opportunity to introduce the latest spatial-temporal omics technology. The title of my presentation is Understanding Plant through Single-Cell Spatial-Temporal Omics. The triple area of BGI, genomics, single-cell omics, and spatial-temporal omics are robust tools to understand biology. And many excellent works have been published in top journals, such as Cell, Nature, and Science. Today, I will focus on the application of single-cell omics and the spatial-temporal omics of BGI in plant studies. Single-cell omics is an important method to explore cell heterogeneity, and it has been widely used to study plant root, leaf, and shoot apex, and so on, to explore the characteristics of each cell types, especially for the real cell types. Firstly, I will start with a single cell study, and that we did and try to answer the important questions that how does colors regenerate into a whole plant? We performed time series single cell transcriptome experiments during the early stage of de novo shoot regeneration. We obtained the single cell transcriptome data of about 35,000 cells at 5 time points. And we found significant cell heterogeneity in colors. We successfully reviewed 7 major cell populations in this developmental process. First, analysis revealed that kinetical regeneration related genes were significantly expressed in cell population 3. And many transcription factors were enriched in cell population 3, indicating that cell population 3 might be an important stage of cell phase transition. And it's interesting, and it brings us to the next question. Where are these cells distributed in callus tissues? Using single cell technique, we could explore the cellular heterogeneity. However, the limitations of single cell studies for plant studies are also obvious, such as we couldn't know the location of cell population for the spatial information of cells were lost. As for the leaf, the main place for photosynthesis in plants, it's mainly composed of epidermis, mesophyll, and vasculature cells up to now. Several single-cell studies have been carried out on leaves. However, for the limitations of SCR-seq, the transcriptomic characteristic of leaf cell subtypes could not be identified so far. There are still limited knowledge about what are the differences between palisade mesophyll cells and the spongy mesophyll cell. Whether the upper epidermal cell and the lower epidermal cells are different. To solve this problem, the development of gene expression profiling technologies with transcript location information is in great need. The previous tissue level resolution of the first plant spatial transcriptome technology hindered the application of it in leaf study. Excitingly, Stereo-seq a new spatial transcriptome technology with subcellular resolution were reported this year. The DNB on the surface of chips contains random barcoded sequence and the coordinate identity molecular identifiers and the poly-T sequence containing oligonucleotides designed to capture mRNAs. The distance between the two DNBs is 500 nanometers, and it's enough to meet the need of leaf research. Applying STAR-seq to plant studies, we partially optimized it and take advantage of cell wall that is a barrier to SCR-seq. STAR-seq plus cell wall staining, the single cell resolution spatial transcriptome technique was upgraded to spatial single cell transcriptome technique. We performed cell wall staining on the same chip to visualize cell-to-cell boundary. After merging and the registration, the cell wall staining image with the exploration image generated by the STOmics visualization system. We then could extract transcriptome information of individual sampled cells based on the coordinate information of the cell wall boundary. This work has been published in Developmental this year. After obtaining the spatial single-cell transcriptome data, we first did the data quality examination by calculating the gene number and the MID number and the percentage of mitochondrial genes in each cell across all 26 leaf samples. The majority of cells have similar gene numbers and MID numbers along all leaf samples, and the gene number was positively correlated with the MID number in each cell, thus strongly indicating a robust and stable RNA capture efficiency of our single-cell method in different sample areas. In total, we obtained about 13,000 high-quality single cells with spatial coordinates. Based on histologic features and their spatial information in the leaves, the cells were classified into 6 major cell types: upper and lower epidermal cell, palisade and spongy mesophyll cell, gut cell, and vesicular cell. To confirm the reliability of the cell type assignment above, we compared the expression level of marked genes of each cell type and also performed PCDG analysis among the 4 major cell types. Here, we show the PC analysis results that the same cell types from different leaf samples were clustered together, while the different cell types from different leaf samples were separately grafted. We further validate the reliability of the 4 cell types by detecting the expression of the previous report canonical marker genes in each cell type. We found that these genes were highly expressed in the corresponding cell types and lowly expressed in other cell types. For example, canonical marker genes for gut cell was spatially expressed in gut cell. This result demonstrates the reliability of cell type identification using the single cell method of STAR-seq. After obtaining the single cell transcriptome with spatial coordinates, how to use the spatial information? Firstly, it facilitate the identification of cell subtypes and the subtle transcriptional difference. For Arabidopsis leaves, epidermis can be subdivided into upper epidermal cells and lower epidermal cells. However, because of the highly similar transcriptomy between cell subtypes, previous SCR-seq data and analysis alone were not able to distinguish these cell subtypes. Using STAR-seq by combining the single cell data with the histological spatial information, we could successfully distinguish upper and lower epidermal cells. And further, we reviewed the biological function difference between upper and lower epidermal cells. For example, the biological process associated with the photosynthesis, cuticle development, or significantly enriched in upper epidermal cells, while biological terms related to response to absence of light was significantly enriched in lower epidermal cells. Secondly, we're trying to explore the cell type specific gene expression gradients from the main vein to the leaf edge. The leaf cross-section was divided into 4 areas from the main vein to the leaf edge. Interestingly, 3 distinct gene expression patterns in mesophyll cells from the main vein to the leaf edge were observed. The photosynthesis process mainly takes place in mesophyll cells, and we found that photosynthesis genes showed spatial expression patterns in mesophyll cells, and these genes were highly expressed at the leaf edge area, including RCB6, RCB 1.2, and RCB 1.3. Thirdly, we reconstructed the spatially resolved developmental trajectories of vascular cells. We performed procedural time analysis on vascular cell and compared the developmental stages in the 4 areas compared to vascular cells in area 1, 2, and 3. A significantly large number of vascular cells from area 0 distributed at a relatively early procedural time stage, suggesting that vascular cells from main vein region were less differentiated in general. We successfully reviewed the complex cell type specific and spatial temporal gene expression features of Arabidopsis leaves. The analysis results have been uploaded to STOmics DB, and it's open access. Using this database, we can easily obtain gene expression levels in various cell types of leaves. Briefly summarize, based on the above results, we have realized the application of spatial-temporal omics in Arabidopsis leaves. We successfully identified the cell subtypes and subtle transcriptional difference. The cell type specific gene expression gradients from the main vein to the leaf edge. STAR-seq enabled the construction of spatially resolved developmental trajectories. Thus, STAR-seq enabled us to capture the in situ transcriptome of each single cell of plant tissues. In addition to Arabidopsis, Stereo-seq had also been applied in many other plants, such as in peanut. Cell type notation is quite difficult for single-cell studies, especially for non-model plant species that lack of the known marker genes. In peanuts, using Stereo-seq, the researchers have successfully explored the cell heterogeneity of peanut root, hypophysetal stem, and pike. This work has been published in Plant Biotechnology Journal this year. There is another case. Stereo-seq has also been applied in soybean nodule. Using Stereo-seq, researchers integrated the single nucleus and spatial transcriptomics captures transitional stages in soybean nodule symbiosis establishment. This work has been preprint this year. In addition, a lot of very important and interesting spatial-temporal omics research are ongoing now. To make the results of spatial-temporal omics research available to our researchers, we're building an encyclopedia of plant spatial-temporal omics research. We and our partners have selected two representative species, Arabidopsis and rice, which represent dicot and monocot, respectively. We will construct spatial-temporal atlas of the tissues and organs of these two species throughout their developmental cycle. And we will also establish a public database for data acquisition and online analysis. We also sincerely invite the participation of multiple partners that could join us and complete the spatial-temporal mapping of multiple plant species. We have also carried out spatial-temporal omics studies on important plant organs and the developmental process. For seed development, including embryo development, seed maturation, seed germination, and so on, we will construct the spatial-temporal atlas of seed developmental process to explore the regular mechanism of SAM and RAM development. The mechanism of source sink conversion in seed matching. The spatial-temporal regulators of seed response to adversity. Additionally, we are also constructing the spatial-temporal atlas of important developmental process to explore the mechanism of cell differentiation and the redifferentiation, the developmental trajectories of bundle sheath cell of carbon for leaves, the development of root nodule, and the mechanism of nitrogen fixation. Previously studied of the developmental process were quite difficult. With the help of spatial-temporal omics, we could change the situation. Since Stereo-seq enabled us to explore the characteristics of plant tissues and cells in situ, review the communication between cells and the developmental lineage of cells. Thus, STOmics would greatly accelerate our understanding the mechanism of plant development and response to stress, and underpin the needs of agriculture breeding research. To further promote the development of spatial-temporal omics, STOC has been founded. ST is an open, collaborative research initiative, and welcome to join the STOC community. This works accomplished by a group of outstanding young scientists. And many excellent partners have also participated in the ongoing research. That's my presentation. Thanks, and welcome to join the STOC community.